Hello, welcome back. Looking at my notes, I realized I forgot to talk about visual acuity on that last lesson. So I'm just going to add that real quick to the beginning of this one before we jump into photoreception. So visual acuity is, you might be familiar with, 20/20 vision, 20/15 vision, 20 whatever. So I just wanted to discuss what that means. So when you have 20/20 vision, this is you, no, yes, sorry. This is you, and this is a normal vision, right? So what that means is you stand at 20 feet and you can see the same thing that a normal visioned person sees at 20 feet, okay? So if you have better than normal vision, 20/15, that means you can stand at 20 feet and see the same clarity and the same detail as somebody with normal vision when they stand at 15 feet. So they have to stand 5 feet closer to see the same clarity that you can, standing 5 feet behind them. On the flip side to that, if you have, say, 20/80 vision, that means you stand at 20 feet. You are always standing at the 20-foot line in this ratio, 20/20 or 20 whatever. And then the normal person can stand all the way back at 80 feet and still see the same clarity that you see at 20 feet. So 20/20 means you're on par with what is considered normal vision. If you have a number that's less than 20 in that second number, so 20/15, 20/10, that means you are still standing at 20 feet, but the normal person has to stand closer to be able to see what you see. If that second number is larger than 20, then that means the normal person vision has to stand further away to see what you see at 20 feet. If you have 2200 with corrective lenses, so you have corrective lenses and the best you can see is 2200, then that is a threshold for being considered blind. So you'd have to have all the conditions for accommodations for being a blind individual if you can't get your vision any better than 2200. That would be you standing at 20 feet and somebody standing 200 feet away. That's what the clarity of the vision that person would have. Okay, just wanted to make sure that was mentioned. So this is the last part of the vision. So this is the second video for visual physiology. It's going to focus mainly on photoreception. I will touch a little bit on color vision and the pathway, the processing of the visual information and the pathway from your eyeball back to your visual cortex and all the stopovers that it does. But it's going to mainly focus on the process of photoreception. So to begin, we wanted to take a closer look at the structures that we're going to be dealing with. So we're zooming way into the retina. So you might recognize the pigmented epithelium, right? So those are the kind of cuboidal cells, have high concentrations of melanin. Here's our rods and cones, right? And then we'd have our bipolar cells down here, and then ganglion cells beyond that. So we're zoomed in just to primarily center our focus on the rods and cones. And then the difference between a rod and cone is the shape of what we call the outer segment, right? So here's the outer segment. We can have a triangular cone shape. We can have a more rectangular rod shape. So that's where the words rods and cones come from. It is in this outer segment that we have an increase in surface area of phospholipid bilayer. In the cone actually has the folds of the membrane in that kind of cone shape. In the rod, they have little flattened sacs, like little pancakes. And so this is going to be the rod shape, and this is the cone shape. But either way, the goal for all of these structures is increase surface area, because what we are putting on all of those in that membrane is this molecule called rhodopsin, this kind of purple dumbbell looking shaped thing. So here's our stacks of membrane, and the little purple dots, that's the rhodopsin. Rhodopsin is a combination molecule. It has a chemical in it called retinol embedded in a protein called opsin. So retinol plus opsin equals rhodopsin, and it is embedded in the phospholipid bilayer. So we can see that. So it's got this big complex protein, and then we have this retinol molecule inside. So that's all happening embedded in this outer segment, right? So all of this is happening out there. The inner segment is basically your general cell part. So we can see nucleus, Golgi body, mitochondria, and then the synapse is the connection with your bipolar cells. Okay. So that's the structures we're taking a look at. The outer segment is where light is actually going to be picked up by the rhodopsin molecule, and the inner segment is the rest of the neuron that then communicates to the bipolar cell. Okay, so we're going to start this process off in the dark, right? So if you're to completely close your eyes, cut off all light from happening, this is what will be going on in your rods and cones. Even though they're only showing rods, it's going to be the same thing with cones. We'll talk about that in color vision at the end. So we have to set up the resting state. Now, we have some prior knowledge from chapter 12 in neurophysiology, right? So we back then, we talked about a neuron having a resting potential of -70, and then when it was depolarized, we did the action potential, and then back to -70. And we had a threshold, right, of around -60, sodium channels closed at +30. So all of that stuff you can go back and review if you need to. But our rods and cones are slightly different because take a look at the resting potential. It's at -40, so it's slightly a little bit more positive than our -70. But also look what's happening down here at the synapse. The neurotransmitter is being released. So at rest, the neuron, these rods and cones, these photoreceptors are actually actively releasing neurotransmitter. But here's the trick, right? Because you're like, well, why would they be releasing neurotransmitter in the dark? Isn't neurotransmitter usually sending a signal? Well, in this case, it's not. It is an inhibitory neurotransmitter. I think it's like glutamate or something with a G. So this inhibitory neurotransmitter is keeping the bipolar cell turned off. Because remember, rods and cones, bipolar cell, ganglion cell, and then to the brain. So if the rod and cone is in the off position, which is no light, it is actively inhibiting or keeping the bipolar cell from depolarizing, which is going to keep the ganglion cell from depolarizing, which means no signal is being sent to the brain. So in this dark state at -40 millivolts, the neurons or the photoreceptors, the rods and cones, are releasing an inhibitory neurotransmitter, keeping the bipolar cell off. Okay? Well, how is that doing that? Well, we need to maintain what's called a dark current. Okay? So in the outer segment, where here's our discs and our rhodopsin, we just talked about that. They're embedded in the outer plasma membrane. We have a chemically gated channel for sodium, and we have a little doorstop molecule called cyclic GMP. So it's very much like cyclic AMP, like a second messenger, right? So we have a whole bunch of the cyclic GMP floating around in the cytoplasm, and it's bound in this receptor site, and that keeps the gate open, which allows sodium to rush in. So sodium is flooding in because it's got this high concentration gradient. It's coming in through the channel. But in the inner segment, if you look down here, we're going to have some active transport pumping the sodium back out. So that's why it's called the dark current, because we have sodium leaking in, we pump it out. Sodium leaking in, we pump it out. And that's what helps to maintain this -40 millivolts, which then allows the inhibitory neurotransmitter to be released. Okay, so this is dark state, no lights coming in. So let's take a look at what happens when light does come in. How are we going to stop the release of the inhibitory neurotransmitter? Okay, so we're going to take a close look. We got to do a little chemistry side note. So you may or may not have had this in a chemistry or biology class prior, but there are a type of isomer molecules called cis-trans. And what that means is it's usually a carbon-carbon double bond, right? So carbon can make 4 bonds. There's 2 right there. And it is going to be connected to hydrogen on each side, and then something else. I'll just say chlorine. I don't know what chemical that is, but I know it as an example from prior classes. So in this case, when we have both of the hydrogens on the same side of this carbon-carbon double bond, this is what we call cis. Okay? So a cis molecule when they're on the same side. If we were to have a carbon-carbon double bond, with the hydrogens on opposite sides, like this is what we call a trans. They're the same molecule because they have the same atoms, the same number of atoms, pretty much the same position. They're just either this way or this way, okay? On the same side, like my thumbs are on the same side, or my thumbs are on opposite sides. So that's cis-trans. So when we talk about this cis-retinol and the trans-retinol, that's all we're talking about. So this retinol that's embedded in the middle of this rhodopsin molecule, the opsin protein, the retinol is inside. In the cis form, it's got a little kink in it, right? So double bonds typically have the kinks. Light comes in, so a photon of light comes in, it's a little packet of energy. It physically changes that chemical bond and straightens it out and turns it into a trans molecule. So cis is in the dark state, trans is after a photon of light has entered and physically changed that protein or that molecule, that chemical. So now we have a trans retinol protein. Well, what does that do? Well, that actually activates the rhodopsin. So we can see in the cis shape, the whole rhodopsin is kind of that paley color. And when it's in the trans shape, it's kind of a darker color. That's just to visually show you that now this whole rhodopsin molecule has been activated. It's absorbed the energy of the photon of light. It is now an active functioning protein. Now in those little membranes, whether it's the back and forth in the cone or the little sacs in the rod, like we can see here, with this activated, right, so this is now activated, it will activate a neighboring enzyme called transducin, right? So now transducin is active. Transducin activates its neighbor called phosphodiesterase. Phosphodiesterase is an enzyme. So remember, we had all this cyclic GMP in the cytoplasm, and we had that cyclic GMP keeping that channel open so sodium could rush in. Well, when you have a whole bunch of activated phosphodiesterase, it starts to remove all of the cyclic GMP and it actually takes it out of the receptor site. Right? So here's what's happening. So all of the cyclic GMP is getting broken down into just regular old GMP. It's not cyclic anymore. So here's the phosphodiesterase making that conversion. Regular old GMP cannot keep that gate open. The gate closes, right? So we go, we close. Sodium can no longer come in, okay? So remember, we were maintaining a dark current because sodium could come in and we were pumping it out. When we are no longer allowing sodium to come in on that outer segment, but we are still actively pumping it out, we're losing positive charges. So what's going to happen to the transmembrane potential when we're losing positive charges? Well, take a look at the meter. We're going more negative, hyperpolarizing from that negative 40 resting state. So negative 70, take a look at what's happening to our release of acetylcholine, or it's not acetylcholine, neurotransmitter. So at negative 70, the photoreceptor, the rod or cone, is no longer releasing the inhibitory neurotransmitter. But what does that mean? If we're inhibiting the inhibitor, we're turning off the off button, that's the same thing as activating that bipolar cell. So when photons of light come into your eye and land and activate that retinol from cis to trans, the rhodopsin gets activated to break down the cyclic GMP. We close that gate in that outer segment, but we're still pumping out sodium, hyperpolarize the neuron, turn off the inhibitory neurotransmitter, and we activate the bipolar cell, which then says, hey, a photon of light landed up there. So then it can activate the ganglion cell, and then that's what sends a signal to the brain. Okay, so here's all of the steps in one picture. I'm not going to go through all again, but you should. As many times as you need to, right? We talked about repetition and memory consolidation, turning short-term into long-term memory at the end of chapter 16. So this is a really important process. So be comfortable describing the dark current, how we get to the cis to trans, what opsin, transducin, and phosphodiesterase does. What's the relationship between the releasing and the not releasing of the neurotransmitter and how that impacts the bipolar cell and the ganglion cell. All of that is good information to be able to be fluent with and comfortable describing. So whatever you need to do, flashcards, write it out, label pictures, talk it with a friend, email me, whatever you need to do, know this process. Okay, so that's photoreception. Now, let's say that's happened. We are blocking the inhibition of the bipolar cell. Bipolar cell is becoming depolarized. It's sending its signal, but we've got to have an off switch. So somehow we've got to turn off this whole process. And we do, but we have to go back and revert everything to how it was in the resting state. And that's going to start with that retinol molecule. So after the excitation, so that's this right here. So here's the conversion of the cis to the trans. This is going to trigger the vision, right? The ability to sense the sight. So that message has already been sent. It's long gone on the ganglion, axons, and all the way back to the visual cortex. If there's no more light coming in on that particular rhodopsin molecule, we have to turn that trans back to cis. So to do that, we actually extract the retinol molecule. We use some enzymes in the power of ATP to make that kink back. We got to change that double bond from a trans double bond to a cis double bond. Then we plop that retinol back into the rhodopsin or the opsin protein, and now we get a nice resting state rhodopsin ready to receive another photon of light to trigger the whole process again. So this is actually not that. It's the blocking of neurotransmitter that stimulates the bipolar cell. Okay. Yes. All right. So this is called bleaching and regeneration. It's bleaching because we're removing the pigment, the retinol pigment out of the opsin. Regeneration because we're going from the trans back to cis and then plopping it back in for a resting state rhodopsin molecule ready to receive. So bleaching and regeneration is this process. All right, so all of that we covered with the photoreception was discussed on a rod, right? So photons of light come in. Well, light comes in different wavelengths. So the wavelength is what we coordinate with colors. So wavelengths here are in nanometers. So nanometers, millionths of a meter. So they're super, super tiny. Millionths or billionths? I think it might be billionths because micrometer is a million, I think. So nanometers anyway, that's super duper small. And the smaller the wavelength, you're on kind of the blue-violet end of this visible spectrum, the visible light that we can perceive in our eyeball. The larger the wavelength you have, you're kind of on the red-orange end of the spectrum. Rods are pretty active, that's the black line here, they're pretty active over most of these colors. They don't really get activated with the photons of the red wavelength, but pretty much all the other colors, you can activate a rod. Oops, just want to erase that. Now for our color photoreceptors, the cones, they are activated in only a smaller range of these wavelengths. So for example, if you're a blue cone, right, you only will do that whole process of photoreception that we just talked about between about maybe 415 and 550. So if a photon of light comes in that has a wavelength of 650, it will not change that retinol from cis to trans. It just does not respond. Only blue cones will respond within that particular range. And again, you can see the same thing for green cones. Let's see, green is between about 450 and maybe 625, and red is between about 500 and 700. Okay, so again, if the photon of light comes in, it's like 425, it will only activate blue cones, green cones, and red cones will not be activated. It goes through all of the same processes we just covered, but they're just specific for that wavelength of light that comes in. Rods basically are just light or no light. They'll react to almost all of the wavelengths. Now, some individuals have difficulty in perceiving color, either completely, which would be a complete color blindness, or different shades like red-green color blindness or blue-yellow color blindness, so there's several other combinations. But what that means is when those wavelengths come in, the cones are not working properly. They're not responding and activating like they would with the different wavelengths. So we have the color blindness plates in labs. You'll have an opportunity to look through those. You may know somebody who's colorblind, or you yourself might be colorblind. What this diagram is, there is a reddish orange 12 in the middle of all those dots. So if you can see that, then you are at least not colorblind to distinguish red pigments or red wavelengths. If all you see is a collection of dots without an actual number, then that likely means that you are colorblind and your red cones are not being activated by the red light that's coming off of the screen. So we can talk more about this in lab since we'll have more of the one-on-one time to do that, but that's the idea of color blindness. It is a X-linked trait, so it is more common in males because females have two X's. So if they have a recessive for color blindness, they have a better chance of having a dominant to override that. But males only have one X and one Y. So if they have the recessive on their X, they only have the Y. That's the maleness. And so they are more likely to have color blindness. It's passed on from the maternal line of the X's that are donated. Well, I guess men donate X's too, but it's just more common in males, typically carried by the females. All right, the last slide that we're going to take a look at is the processing of this information and the visual pathway. So all of the photoreception that we just talked about, it would be like a photon of light, and it's usually not just one photon, but let's just say one rod gets activated. So let's say this rod gets activated here, this guy right here, it's going to come in and it's going to maybe communicate to this bipolar cell, which will then communicate to that ganglion cell. But the bipolar cell might have a threshold where just one of those rods is not going to be enough to activate the bipolar cell, which would not be enough to activate a ganglion cell, so that information would not be sent to the brain. So it could be that all of these in this receptive field have to fire before the bipolar cell can be activated to activate the ganglion cell. So there's a couple different types of these receptive fields or these ganglion cells. So in the notes, it talks about M cells or magnocells. These are the ones that are going to be kind of more on the periphery of the retina. They are monitoring maybe upwards of 1,000 rods. So your receptive field has 1,000 rods in it going on to a fewer bipolar cells, all those bipolar cells being monitored by one ganglion cell. So if a photon of light comes in and enough of those photons activate enough of those rods, we might say we can perceive light in this field of view, but it's not clear, it's not focused because we're monitoring so many rods. In that fovea, remember the little dented area off center on the back of our retina, those are monitored by parvocells, which is one ganglion cell for one bipolar cell for one rod or cone. Mostly cones are in that fovea. So that gives you a much better precise vision. So that is the parvocells and the magnocells and this receptive field. That the more area of focus, which is in our macula ludi and the fovea, the fewer photoreceptors are monitored by a ganglion cell. In kind of the periphery, the edges of our retina that are on the side, kind of this peripheral view, that's going to be our magnocells, where we have way more rods and cones being monitored by a single ganglion cell. And then here's our horizontal cells and our horizontal cells and amacrine cells. They just help to kind of regulate and intermingle, what's the right word I'm looking for? Interfere is not the right word. Facilitate or inhibit these signals as they're coming from the rods and cones to the bipolar to the ganglion cell. So a lot of convergence and processing of that information before the signal actually leaves the eyeball through the optic nerve. Okay, so then our visual pathway. So our eyeballs have all of those ganglion neuron axons are what make up the optic nerve. And so if I'm going to track the information that's coming on this side of my eyeball, it is going to stay on that side of my path and eventually go to that side of my brain. So this is my right eyeball. The right side of my right eyeball will stay in my right hemisphere. The left side of my eyeball will cross over at the optic chiasm and go back to my left hemisphere. So each eyeball information splits to go to each hemisphere. So we're receiving both left and right information on your left and right hemispheres. That's at optic chiasm. Remember, that was one of our landmarks from our brain anatomy section. So we get a little bit of, we get the crossing over, right, the optic chiasm, kind of right by our pituitary gland. Some of the locations of stopping over, so this will be a refresher from our brain anatomy, is we have our suprachiasmatic nucleus, brainstem and diencephalon. We have our superior colliculus, which was part of our visual reflexes. Okay? We have our lateral geniculate nucleus, which was part of our thalamus, right? And then here is our cortex, and then our association areas, right? So making sense of that raw data that's coming in. All right, so that is vision. I think we covered all of the physiology that we needed to cover here. Then the next couple videos are going to be on ear anatomy and then equilibrium sensation and hearing sensation. And that will wrap up our special senses. Okay, bye.